We've reset our system. We've been having some technical difficulties, both on our end and on the other ends. But hopefully it's resolved now. We are just at waypoint 4 out of 17, in case you're interested. And here we just fired 2 Niskin bottles to collect some water samples. And we are about to do heat flow measurement and 8 push cores at this site.
Yeah, we're going to do the push cores first. Yeah, no, I'm just showing you that the probe went in. Oh, I see. Yeah, yeah.
Mm-hmm. We didn't forget to say woo. We did have a really good tack with him sashimi in his house, so do you remember? After
That was a good party. And then it continued.
Yes, exactly. Yeah, yeah.
Almost getting cleared.
Do 75 first. So that these guys, this one first.
trip was attacked me 9 years ago.
No.
So as people wander back in, thank you for sticking with us through some of the connection issues that we've been having. It's always a challenge sometimes getting things stable. We showed our connection was getting a little flaky on this side, so we got that fixed. And then YouTube was still saying, it's a little flaky. So we just decided to restart the whole thing and get an early part 2 going. We'll see what that does for our timer in a little bit. But for anyone just coming back in or just joining us now, we are settled down and we are doing, or we're getting ready to do some push cores here. We're just waiting on a little bit of sediment. And you can see Adam is currently pulling the course up and out. And we're going to be pressing these down into the sediment. For anyone who's never seen this before, right, this clear tube is actually going to get pressed down into the sediment, just like this. Oh, very nice. It's a satisfying thing, I feel like. But that's going to get pressed down in there, and then we're going to, are we doing all 8 here of the push cores? Perfect. So we're going to punch all 8 of these in. And then go back, and when you take them up like this, they bring a nice little chunk of sediment with them. And you can see, even within that, since it's a clear tube, you can see a little bit of layering there. And there's all sorts of projects on board and onshore that'll end up using this sediment. Everything from looking at the actual microscopic structure of the sediment, the little skeletons, the sea critters that make up that sediment, the minerals that make up that sediment, and even the live organisms, micro and macro, that are living in that sediment. So this is actually, you know, it seems weird, right? We've got this robot on the bottom of the sea kind of playing in the dirt. But it's actually very important, and we can get a lot of information about the region from these push cores. See a question about how the ship gets its internet, and we do use satellite internet. Obviously, we can't have a cable run to the ship. But we have several different networks that we're connected to, and that way we can kind of split it out. So this streaming is on its own network, just so that it doesn't get overwhelmed if someone else is doing data use somewhere else. And then the crew and scientists are on another network as well. And of course, we use satellites for a lot here on the ship. We are using satellites for our navigation as well as our internet. So when you're out at sea, right, you can't really be wired up so easily. So satellites are where it's at. And obviously that's something in the news nowadays, the expansion and more accessible nature of satellite internet. And something that interests me about that is that it'll maybe make it a little easier for groups like Schmidt Ocean Institute to bring these deep sea operations to people around the world.
Huh.
Oh, having an issue with our grip here. Well, I guess
No,
the issue isn't the grip.
Yeah,
Yeah.
grip looks fine.
Yeah. There we go.
I think what I'll do is I'll have to put this one in the mud and re-grip it so that we're not grabbing the mud. Re-grip it and then re-hand it out. This is not a core yet.
Yeah, don't log this one yet. Yeah.
often have you looked through like sediment cores?
Very often, I actually find it deeply therapeutic, which perhaps says something about
Looking through
me.
the cores.
Yeah.
Yeah.
So in the top, most of the animals tend to live in the top 10 centimeters, lots of small animals. And so we get the cores on deck. We then slice different sections of them. And then we're not doing it on this cruise, but on other cruises I've been on, we sit with each section and sort through it using forceps and microscopes. And it's, yeah, there's just something very mindless and therapeutic about just working your way through a sample and pulling out all the little animals there. And there's often, that's where most of the diversity is in abyssal sediments. So you're not getting a huge amount of animals, but every single one is often a different species.
Yeah, that's kind of given me like thoughts of when students do owl pellets in school, and you're
Do
kind
what?
of digging through owl pellets.
What's that?
So like when the owls swallow their food, right? If an owl's out there eating voles or little shrews or mice, whatever it is, they digest the meat of it. They can't digest the bones, and rather than passing that out the rear, they'll actually like cough up a bolus.
What?
Yeah, and you can actually purchase these.
Oh my gosh, from
And
where?
it's an activity in schools. I know that like there's actually farmers that have,
Like resident owls.
yeah, they have owls that like live by
In
their
their
barn, and they
barns.
just collect them and sell them to science supply companies.
And so they just find, you just find them on like the floor? The
Yeah.
owl's just like that.
Yeah, yeah. Underneath where the owls roost, you'll find like these little hacked up boluses, and
How big are they?
little. They're little. For people at home, picture like a very large hairball from a cat.
I was gonna say, does it look hairy?
It's hairy. Yeah. Did you do one, Adam?
Yeah.
Yeah.
I did a couple of those in school.
Yeah, I never had an opportunity to do that in school, but always seemed very cool. And so it's a student activity frequently to take those and break them apart and look for the bones of the prey, and then you can ID the prey. And I've actually led similar activities using like sea lion scat to show students
Oh
how
my
scientists can learn the diet of the animal from the scat. And I know that I want to say Midway, you can actually get boluses from albatross that are at Midway. They only send like one per class though, and you're supposed to kind of reuse it and stuff. But that's to look at sort of what's being hacked up by the birds on Midway, the albatross.
Like maybe the island.
Yeah, Midway Island. And so when you were saying, you know, that you would kind of sit around and with forceps go through these sediments, that kind of gave me that forensic flashback of really getting in deep with this. And I assume you're kind of like counting how many little worms you're finding. And does it tell you, I mean, I guess what does it tell you, right? When you're looking through these things, what sort of thing are you typically looking for?
I mean, so you're pulling out any animals that you're seeing, and it can be everything from polychaete worms to Tannaid crustaceans. I think the main groups tend to be polychaetes and crustaceans, but we do get other things. And it's essentially like a smaller view, if you will, or a different view of what is living in this place. So everything we see on these ROV dives is just one size class of animals. They're the biggest things, the things over like a centimeter in size, roughly. But there's so much more life in deep sea systems, things we call macrofauna, myofauna, and then of course microbes, which we're here on this expedition to look at as well. And so using these cores is how we're able to look at things like macrofauna and myofauna and basically understand the different animals living in the system, and then work out all kinds of stuff from those animals, like how they, what their roles are in the ecosystem. And many of them play a key role in like carbon cycling, for instance, and carbon sequestration or nitrogen cycling. So there's a lot that these smaller animals do that are critical to the functioning of these ecosystems. And by doing these cores and working out what's there, that's the first step in being able to answer those bigger questions.
Okay. Sometimes we get folks asking why there's lids on these. Obviously, in our coral quivers and our bio box, the lid is to kind of keep the critter or the sample secured down in there, but these seem to be a little more secured by themselves. But if we're taking a sediment sample that has like very gaseous sediment, has maybe like methane in it, if we don't put this little bungee cap on, as we're ascending, that gas will expand and it can actually push the core tube up and out of its holder. So that's why we have these little bungee caps. Just a little bit extra security.
So we can also use these cores to have a look through through time, essentially. By working our way down the core, we're able to move through thousands to millions of years in time, potentially. And so that's one of the things that geologists on board are going to be doing once they get back to shore.
That's something I thought of when we were on our very long, the 30-hour dive, and we found these cliffs. And I mean, it was like 10 meters of sediment. And just thinking about taking a sample of the base of that cliff and what sort of age we would see on that sediment.
Halfway.
Oh. Kind of an interesting question there, Diva.
Yeah, and luckily Beth is here, and so I can ask her when she's finished.
We'll catch her when she's done. Yeah, she's in the back discussing some dive plans. But for anyone maybe unable to see the chat and you're just watching along, we're going to dig into this question a little bit, hopefully. Obviously, the larger samples that we bring up, those animals frequently don't survive the trip up. Or they aren't in the most, like, I'm trying to think, they're not the most active when they come up. The change in pressure does obviously impact these animals. And so when they come up, a lot of times they're not like super vigorous or anything. But we had a question about whether the fauna and organisms, the microorganisms in the sediment samples, if those were still kind of wriggling around when they came up. I don't know.
Is Wilder. Huh.
I feel like microbes could probably be cultured off of these sediment core samples, but I don't know.
So yeah, so Miguel was just saying that yeah, and Beth, please jump in, but that many of the microbes that we collect here in the smaller animals do stand a better chance of making it, but microbes, especially like around the majority of them, tend to be have wide tolerances of pressure, but and a much smaller percentage of them will very specifically need the high pressures that exist in the deep sea to survive. So, does that sound right to you, Beth?
Yes.
Yeah.
Especially at these depths.
Are you doing any culturing? No. On board? No.
No culturing currently. That was a cool question, though.
I can figure that out rather than okay, perfect.
I just say this is fun.
Oh, perfect.
There's always the chance of writing it wrong.
Ooh, monopsid. Isopod. Walking to the side of the cores. Another microbe question we've unleashed. How much success is there in culturing these microbes? Are there just a few that seem to dominate the plates? I'm just thinking of the Great Plate Anomaly issue.
You want to answer that, Miguel?
How much success is... Oh, sorry.
Okay. Get on in here. Yeah, so every teaspoon of this sediment probably has at least 1 million to 1 billion different microbial cells in it, and probably thousands to tens of thousands of different quote-unquote species of bacteria and other microbes like archaea. And typically, trying to culture microbes out of this, you kind of commonly get the same things growing over and over again, the weeds, we call them. And they may not even be very dominant in the system, but they're microbes that like to, whoa, that's a big fish.
I was about to say, big chimera.
I know it's too deep, but I'm just like,
Okay, sorry, go ahead.
That's fine.
Yeah,
You know, we'll break for fish.
chimera.
Um,
Chimera.
So there's some microbes that perform well trying to grow in isolation, but we think actually a lot of microbes prefer other microbes around to grow. And so the idea of getting them into isolated culture doesn't work very well. We have probably less than 1% of all bacteria, far less than 1% in culture. So our only ways of really knowing who these microbes are is by extracting their DNA and sequencing it to figure out their genetic diversity, because trying to cultivate them is very difficult.
Thanks, Beth.
Do you have which percent you know? Thanks. Okay, maybe I'll take some microbe questions for now.
Okay.
thank you, Beth.
So you may have noticed the rat tail or macrourid passing behind the ROV. Looked like a pretty sizable one. So fish are a really good example of animals that don't make it to the surface because many of them have swim bladders that have air in them, and with the change in pressure, that air expands. So yes, any animals that contain air with them usually do not make it to the surface.
Yeah. Looks like we've got one more push core after this. And you can see here that the sediment is pretty sticky. It sticks even to the outside of the cores, and that obviously helps keep it inside. I have seen like sandy bottom core samples before, but those, I've seen them on deployed landers that push the core down, and then as that lander takes off, a little cap sort of slaps on the bottom, a spring-loaded cap. The sand or a less sticky, less viscous, I guess, sediment wouldn't stick in these cores as well.
Wonderful.
Nice.
Ooh, jelly in front.
Yeah, let's see what we got here.
Quick zoom.
Just down on the ground.
There's a jelly just off the front of the basket, which I think you should now be able to see on the live stream, hopefully.
Yeah, yeah. Just down there, little pink jelly.
Ooh.
The one on the right is 5. It crosses.
Yeah, the one.
Yeah.
Yeah, the one going like that way.
So today our dive plan is to move, as we have often done, across the abyssal plains to the foot of a seamount, and then we're going to begin to climb the side of that seamount. And then when we get to an area where there are thought to be some quite steep and rocky surfaces, we're going to move horizontally along those. And as always, we're going to be looking for areas of shimmering fluid, any evidence of octopus brooding, whether that is individuals themselves or eggs or octopus egg cement, remnant octopus egg cement. And if we don't find any of those things, then we're going to be observing and collecting many of the other species that we may see on this upper edge of this area informally calling Caballito. And the area that we are diving on today, even though we dove here yesterday, it was on the southern end, and so this area that we're working in today, again, no one has ever seen before, no human has ever been here before. So it's exciting, whatever we're going to find.
I'll toss up the map real quick here. You can see our
Thanks,
plan.
Alex.
So we're at waypoint 4, still down on the plain there. But you can see we're going to make our way up pretty quickly here.
Right, so now that we've finished our push cores, we are going to do a heat flow measurement. How are you feeling? Ready? This amphipod doesn't have a partner. So right now we're looking at one of these, while we're setting up for the heat flow, we're looking at one of these worm tubes that they build out of the sediment. And then on it is an amphipod. And this amphipod doesn't, they're usually here in pairs, but this one is not. So I'm not sure what that's about.
I think so. I tried to back off the verts as much as possible until we started to
1
float.
and 5.
Okay, thank you.
Yeah, 1 and 5 on the distance. Yeah.
Okay. Okay, I'm recording.
Ready?
Yeah.
Coming down.
Ah.
That's all we get. That's
What?
You getting that last there, Mr. Rob?
No, I wouldn't think so.
I'm not getting 4 either, so.
Maybe we should,
Okay.
yeah, another spot.
I think this is gradually up. So I don't know if there's, we just lift up and fly like 10 meters forward and we'll turn 90.
Turn 90 to the
Yeah.
port. Yeah.
Pull those cores into me, grab, pushing us forward or something. Actually, what I'll do is I'll have to remove. So we're gonna cancel that one, Rob, is that okay? Yeah, that's right.
Coming
All
up with
right,
probe.
so we didn't fully get the probe in. You can see it kind of stopped above the bottom there. And there are several layers to the probe or sensors along the probe that they need all of those in so that we can get a gradient. So the pilots are currently working to reposition us. So hopefully we can get a little bit better deployment of the heat flow probe.
Bye. Who's taking over for me? Rachel.
All right, it's got a little bit of a handoff happening here.
So I am going to be handing over at a good time as we're moving slightly to get a better position for our heat flow measurement where the probe is able to get fully within the sediment. This one we met some resistance at the bottom. So I'm going to be handing over to Dr. Rachel Lauer from the University of Calgary, who is here on board with her student, Rob, to do these heat flow measurements.
You're leaving us.
Leaving you in the throes of excitement.
Yep. So that gets one.
What do you think?
Which way's up there? Right there.
Yeah, coming down. Oh yeah.
Yeah, that's closer. Yeah.
We're already in.
No. That looks way better. You just want me to still probe? Yeah, if you think it holds stable. Yeah. Okay. Let me know when you're stable.
Okay. Ready? Oh, buds.
Abyssal mud. Rachel's not here yet. It's still me, guys.
For anyone just tuning in, we are currently setting up for a heat flow probe deployment, but we are 3,140 meters below the surface at Caballito Seamount again, except I know there was a debate as to whether or not this met the height requirements of a seamount. I don't know where we are. But we are down in the plain, and we're going to be working our way up the slope here. And anyone who's never tuned in for any of these before, right, you are actually watching live footage from a remotely operated vehicle named Sebastian in this case. But think of it as a kind of a cross between a submarine and a Mars rover, but we're not sending it as far away as Mars. And since we're only sending it, I say only, but since it's a few kilometers below us, it's actually able to be tethered back to the ship. So we have pilots here on the ship named Falcor 2. They're actually able to pilot this robot in real time down on the sea floor. So all these little manipulators, the hands, the arms, the claws that come into the screen, those are being controlled by the pilots here, as are the cameras, and obviously the vehicle itself is being flown around. And so this sort of instrumentation really allows scientists to pick their specimens, their sample sites, how many samples, which particular branch of a coral we take. ROVs allow us to be really, really selective with that, and it's very useful. The other big thing about being a remotely operated vehicle instead of a human-occupied vehicle, we don't have to worry about Sebastian getting oxygen or getting, needing to go to the bathroom. So we can actually keep this ROV in for quite a while. Now we're doing about 16-hour dives right now, but earlier in this expedition we had one over 30 hours in length. So ROVs are just a very powerful tool for deep sea exploration. And for anyone just joining us, we love to share that exploration with you at home. So, we hope that you'll enjoy exploring with us.
Yeah, like I think you're like you said, like there's no chatter at all. Like you're like fully immersed, right? Like even the RAM essentially, like the assembly of the RAM. Yeah. So you're actually probably this much in the sediment below. Right. So maybe RAM something to stabilize.
Question of my shift change. Yeah, you'll occasionally hear me hand off to people. I am the multimedia technician on board for the expedition, so I usually work the daytime hours, and I'm on stream a lot here, but I also step away, obviously for meals and little breaks and that sort of thing, but also one of the other things we offer are shoreside connections to groups of students or we can do like a summer camp could call in and that sort of thing. We've been working with students this expedition and making connections back to shore. So I also go and manage those. And then I also, after these dives or during them, if I have some relief, I can pull highlight clips from these dives as well. So that's what I do. But I do get a lot of help from the marine technicians on board. Who are also in charge of things like the CTD casts or running the bathymetric gear on board, getting the nice maps for the scientists. The science team, or the scientists on board, you hear them kind of go through their rotations. And when they're not in here, they're maybe looking at samples that have come up in an earlier dive, crunching data, that sort of thing. And then the pilots, obviously, their job is pretty demanding of them, driving around this large robot and carefully collecting samples. So they kind of get a couple little rotations through the day, just so there's a bit of a break for them there. So that's kind of what the day looks like.
Almost escaped.
I hope so. I would ask more, nothing less of you. Well, I'll be measuring it
Good morning, everybody. This is Rachel Lauer from University of Calgary. I'm here with Rob Perrin, also from the University of Calgary, soon to be Dr. Rob Perrin, working on the heat flow measurements. And we are joined by Wendy Matamoras Calderon, who is taking notes of all of the things we're doing and capturing all of the animals that we're seeing. So we're just started a heat flow measurement, which obviously is very exciting for several of us and maybe not everyone, but in the meantime, what we like to do is zoom in on little critters like this guy. See spider? Yes? No? Okay, see, I'm 70% of the time I'm wrong with my biology. So I'll stick to what we're doing on the heat flow today. This is our second heat flow transect on this outcrop, previously referred to as Caballito in at least one paper, so we're tentatively referring to it that way. It has no additional measurements that have been collected on it prior to yesterday. We did do a few multi-beam tracks over the outcrop to further refine our understanding of the topography or the bathymetry around it. And it's 2.5 kilometers west of the outcrop where we were yesterday. We know that outcrop is discharging. It's slightly smaller in terms of its surface area and height. They're both technically outcrops, but the one east of us is definitely recharging. And the question here is whether this slightly larger outcrop is functioning as a recharge site, or more likely it's doing a little of both. The larger outcrops tend to be doing a bit of recharging and local discharging. So some of the water goes in the outcrop, warms up a wee bit before it makes its way to an exit point. And a larger portion of that water, if it's a recharge site, will make its way into the crust, where it will head away from the outcrop in all directions. And ultimately, if we think about a pair, it will come out a discharge site. So one of the questions we'd like to answer, but really don't have enough time or geophysical data to answer on this cruise, is whether or not these two are a pair that is acting as what we call a hydrothermal siphon.
So during these heat flow probe deployments, we have a nice opportunity to look around closely in the sediment, which is always fun. Notice all the little things that you just don't notice when you're cruising above it.
So our transect yesterday was on the east side of the outcrop. We started in the abyssal plain, looked very similar to this. Lots of sediments and very little else. Although if we zoom in, there's always life at this seafloor. Today we are wrapping around to the north, again starting from furthest point from the outcrop and conducting these measurements spaced about 100 meters apart along a transect. And the idea is we want to see if we can capture what the background heat flow is, as well as the heat flow associated with the outcrop. So ideally those first couple measurements will give you a sense of what the background heat flow is in this region. And we know from previous work that most of the heat in this region, 14,500 square kilometer region of the seafloor that has 11 or more outcrops, is about 30 milliwatts per square meter. What it should be based on the age of the crust and the fact that it cools at a known rate is about 110 milliwatts per square meter. So about 70% of the lithospheric heat has been washed out as a result of vigorous hydrothermal circulation. In order for that much heat to be missing or removed through this process requires very high crustal permeability. In fact, the highest crustal permeability that has been measured in any part of the ocean crust. So this is a really unique area, and we are currently at the southern edge of that chilled region. There's a thermal transition to the south where we go from chilled crust to crust of normal heat flow. And when you cross that, it goes from about 30 milliwatts per square meter to 110 milliwatts per square meter. And that's just to the south of the outcrop we are currently approaching. Yeah, so just to think a little bit about how previous measurements have been done, we do have transects of heat flow conducted in this area, not on this outcrop. None of them were focused on this outcrop. And those are over-the-side measurements using a much longer probe. And the advantage of that is you're just sampling more of that geothermal gradient. So you have more sensors, you have a longer sensor to assess the gradient. So you have a higher resolution. The smaller probe is something that can be deployed via an ROV or a human-occupied vehicle like an Alvin, submersible, and allows us to do very similar types of measurements using obviously fewer thermistors, fewer measurements, but our estimates are very consistent with those longer probe measurements. And I do think, yes, this is a very important part of our understanding of our ocean. This process of hydrothermal circulation, where water moves in and out of the crust, is responsible for overturning our oceans every 100,000 to 500,000 years, which sounds like a long time, but as someone who thinks about geologic time, that's not such a long time. And what that means is it's really impacting ocean chemistry, that interaction with the crust. In different places, the residence time of those fluids is much longer because they're lower permeability. So thinking about Juan de Fuca and other studies in that region off the coast of Oregon and Washington in the United States have focused on much younger, warmer crust, and conducted experiments there to understand how fast fluids move. And the punchline there is it's about 3 orders of magnitude lower permeability there than it is here. Bless you. What is that?
Yeah, the isopod there is reaching very high up.
He's got a question.
He's got a question,
I
exactly.
have a question.
Yeah, they're cool. We see these swimming around sometimes. Different types of isopods with these long, long legs. It's just gonna tiptoe on slowly. Small enough, we're gonna try and get the manual focus on it. There we go. On the move. Late for work at the deep sea. Now it's interesting when he has those legs up. You can see there's a little more of a clawed tip to it, and that does seem to sink down into the sea floor. But then, kind of the white portion of the leg just stops. So, it's got a little bit of a grip. Yeah, he's fighting our focus.
Oh yeah, that's good for that one.
All right.
All right.
Probe coming up.
Heat flow probes coming up out of the bottom. We're going to be moving on.
That is still on.
Right as he settled down. It is what it is.
Oh,
Nice shrimp swimming by.
big shrimp.
Okay, lifting off. Going to 5.
All right, so we are moving on to waypoint number 5. Which is just at the base of the slope here. I'll toss the map up one quick time again.
Okay.
So we're leaving waypoint 4 and headed towards the slope here.
So we're going from 45 and then after this one, then
You can see the two scale lasers there, the two green dots on kind of the lower third of the screen there, a little above. And those are 10 centimeters apart, and they allow scientists that are reviewing this footage to kind of figure out the scale of the image and the scale of anything we're passing over. And you'll see oftentimes we'll kick those on when we first see an interesting specimen as well, so that we can kind of measure out the size of it. Little jelly drifting across the seafloor. We've seen some comments about all of the xenophyophores. We have seen a lot of them, not just this dive, but this expedition. But as we're cruising along here, you might occasionally see something that it's kind of colored like the sediment, but it almost looks like it has the texture of like a round shower loofah or something. A weird little brain. But those are xenophyophores, which are unicellular organisms, despite their size. I do think they're multinucleate though, so it's one cell, but multiple cell nucleus is there. And we have seen a lot of them.
What is that?
That's really bizarre.
Yeah,
What's what?
those little
Oh yeah.
Little pock marks.
crop circles,
Yeah. Little marks.
footprints.
It's fun, right? There are tracks.
Yeah.
Yeah, things leave tracks down here, and if there's not a good current, it doesn't get smoothed out very quickly. So, all a matter of sign and spore.
Deep sea gopher holes.
Question about how high are we above the sea floor? So our altitude, where it's not a lot.
0.99.
About a meter.
Yeah, just shy of a meter. Or no?
Yep.
Yeah, okay.
There's 6 degrees C where it's 0.9 degrees.
Okay. Yeah, there's a screen here, and it's showing 0.9, so just shy of a meter, which can be a little deceiving, the camera angles. We've talked about this, especially when we're going like down a slope. It can look like we're very high up and the seafloor is very far below us. But if we put on like a camera that shows kind of the rear of the ROV, you can see that the rear is very close. It's just that the ROV doesn't tip. It doesn't tip to follow the slope. We always kind of stay level. little tripod fish there maybe, or spider fish off to the side, but we're booking it. We're doing a transect to the next waypoint. So scientists will review this footage and they'll use those green scale lasers that I talked about to kind of take a survey of what we see and the sizes of things.
We're going fast.
I thought this was a non.
Oh, no, it's just like hyperdrive.
Yep, constant speed, heading, altitude, and you get those laser dots on, and that kind of gives a nice uniformity to the area on screen, so scientists can really sort of figure out a density of what they're seeing. Looks like we've got quite a few new people jumping in the stream here. So if you're curious what you are seeing, this is live footage from a remotely operated vehicle, currently 3,130 meters below the surface of the ocean off of Costa Rica. And we are headed towards what we had called a seamount, Caballito. And currently just transiting across the plains next to it. And I'll toss the map up one last time here real quick, as we are almost at waypoint 5. So you can see our trail of waypoints, and we're going from the purple towards the slope. And we're going to work our way up the slope and then kind of hang out up top for about half of these waypoints as well.
What are we doing when we go to 5?
What's that?
What are we doing when we get to 5? Stabbing.
And along the way, we're stopping, we're taking various samples and readings. Once we start heading up the slope, we're hoping to find some venting of warm fluid actually coming up out of the ground. And for that, we're going to be looking for what we call the shimmer or shimmering water. It kind of looks like sort of the shimmering air above like a hot street, or you know, if you've been running your car for a while, above the hood, you might get that little bit of heat waves. We see that in the water, and that kind of tells us that there's some sort of venting going on there. So keep your eyes peeled for that. We certainly will be here. And we're very glad to have folks along exploring with us.
Ooh.
Let's see. Spaghetti. Chicken breast.
Tall stock that we just passed. That tall stock we just passed, you can see it on the heat probe cam there. So we're only just above that.
To see, like, might stay up for dinner.
Yeah.
Think about it for the day. Like, nah.
Should I go all in on lunch?
Yeah, exactly. Yeah. You know, you get it.
I get it. Yeah.
It's the first rodeo.
Yeah. It's nice to know in advance.
Very much so. I always did find it annoying when the ships would put the menu on.
I know.
Yeah.
Yeah. This is where it should start going up a little bit. In between this one and the next one. Yeah, because you can see the edge of the contours. So we're sneaking up on the location of waypoint number 5, where we will do another heat flow measurement. And it looks like in between 5 and 6, we sort of hit the base of the slope of the seamount, and should start getting a little bit steeper after that. As mentioned, we're looking for on this dive, in addition to just everything that we see, because we haven't been here before on this particular side of the outcrop, is potential evidence of fluid venting. And even at a couple of degrees above seawater temperature, which here is, where is it?
You got it there on your screen.
Oh, it's on my screen. 1.85. 1.85. So even 1 or 2 degrees above seawater temperature, you will see a little bit of shimmering. And it might require a zoom in to really be able to see it. So once we do hit the sort of the rocky outcrop, we will be taking a few pauses to try and make sure we don't miss any potential vent sites. Oh, there's one of those acorn worm trails in front of us. See, I got one. Acorn worm. I did not know what an acorn was before this cruise, so learning. Always learning. This is a great opportunity for people like me who think about physics and math and modeling to interact every day with people who have an incredible understanding of these systems and these creatures and their behaviors and morphology and Latin names that I never remember, but yeah, it's a truly transdisciplinary cruise, which is amazing.
All right, coming in for a landing. Nice and easy. Kick up all the mud.
It's a little hard, huh? No?
It's like more clayey,
Yeah.
but I didn't hit anything hard.
Okay.
There was resistance going in.
Yep.
So
I think we're fine. Yeah, we have a
Yeah.
smaller diameter.
yeah, and it's on a hydraulic ramp too.
Yep.
You just may want to wait till this turns out a little.
Yep. Cool.
That was the only thing.
Awesome.
What do you think? Is that clear enough? No. You barely see it. So.
Yeah, it's
How did we kill that with the light, which is yeah, it's kind of good to see it.
Yeah, so we're waiting now for the camera on the heat flow probe to clear up a little bit. It's cleared up quite a bit in front where we're using Excalibur to stabilize while we do a measurement, but just need a bit more time so we can make sure that the probe is safely inserted into the sediments, doesn't encounter any rocks on the way down.
Good thing too is that we launch. We'll see what it'll be look like. How you feeling about that, Rachel?
Yeah.
You wanna try it? All right.
You ready, Rob? Well, let's make sure Rob is.
Yeah, recording.
We're good.
Coming down.
Oh yeah.
Well, that's in there.
That's in there.
Yeah.
All five. Sweet. All right. Got a good heat flow measurement started. In case you haven't heard me say it on previous days on the mic, these measurements take about 15 minutes, and there are two parts to the measurement. As the probe is inserted into the sediment, there is some frictional heating. As that decays, after about 7 minutes, we get a sense of what the geothermal gradient is, or how heat or temperature is increasing as a function of depth. Then at about 8 minutes, we fire a calibrated heat pulse, so a known amount of energy is injected into the subsurface, and the decay from that, the thermal decay from that, allows us to estimate thermal conductivity. And the product of those two is heat flow. So both of the components needed to estimate heat flow can be obtained through a single 15-minute measurement. Wow, that guy is cool. I want a picture of that.
So what's that thing called?
Anyone? Wendy, do you know what that's called?
Sorry?
We're consulting with an expert who's on the ship through WhatsApp. Get back to you in a moment.
Okay.
But we have a picture of it, and I'm sure she'll be able to identify it.
But it's a type of coral, right?
Yes. You're asking a geophysicist or a hydrogeologist.
Sorry?
Sea pen. Okay. A ginormous sea pen. Huh? Oh, I don't know. Send Beth a text. Ask her. I am not El Jefe.
A grand fromage.
Exactly. Grand Poobah. One, two, three, four. There's 9.
I don't have my glasses, you can see anything. The video of the waste.
Yeah, it's 9 branches.
Very nice. Así se puede.
Okay.
Yes, but the stalk is so long, but I would be interested in a piece of the stalk and the whole upper part.
Okay.
And maybe a part that you think that could fit in the quiver.
Yeah.
So like
Just
10 or centimeters.
10
or
centimeters below.
centimeters from the base, maybe.
Yep. Do we want, you guys hear that? I'm just verifying that.
And we can
That's
call
it.
it.
Yeah, okay. Alright. So I guess the plan is to try and get this guy, but about 10 centimeters of the stalk from the top, because it's the stalk is very rigid. We won't get it in anything. It'll probably struggle with it. So it's possible to do it, collect it there.
How much of the land, the edges, you can get like 20 centimeters maybe?
Yeah, so somebody broke the lasers on it.
There's some interest in collecting this sample, and we're talking about the best way to do that based on the storage we have on the porch of Sebastian, and as well as understanding the sort of the physical properties of the stalk itself and how rigid it is. Yeah,
Can we use the barrel box or no?
Not- I don't think so. No. Is it something that would go into a quiver?
Fit in the quiver.
You can make it fit.
Okay, make it fit.
That's the right answer.
Don't destroy it, but make it fit.
I won't destroy it, but I might have to give it a little
Push. Yeah.
knock now.
Okay,
Yeah.
okay.
Gentle, yes.
Thank you. Okay.
Nice.
We'll snip.
It's tough work. Can we get a? It's beautiful.
Now I can see it.
Hmm.
Okay, that's good for that one.
All right.
Pro coming up.
I think what I'll do, because you're probably gonna do one more heat flow? if
We
can?
will, yeah, I mean, I think we'll see about the sediments, but if we can, we'd like to, yeah.
So I'll just, probably gonna have to reposition to get this guy.
Yep.
So I'll just pull up and then we'll just kind of set him there, and then I can grab him again without him direct.
Yep. Cool.
Cool. Ooh. All right, so you want to see where we went? So we're at 5. So between 5 and 6, we're doing the third transect. It's very likely that this we'll see about getting.
Okay.
But Oda requested that guy.
Oh, okay.
So we're gonna
All right, popping up over there.
about 10 centimeters below the top
Right.
and put it in full there. So let's get you there. Get in there. Exactly. Yeah.
And then transect after that, and then maybe a heat flow.
That's right.
And then
not
probably
transect that.
anything after that, probably just exploring.
Yeah,
so
it's-
look deeper.
Yep. And from that point on, it's opportunistic. Like one of us will be in here, but
Yeah,
just check.
just happen.
Yeah.
Here? Okay. I'll just sit down. Okay.
I'm just going to sample this. I got the Excalibur, so I'll just put it in the dirt and then we'll wrap it up with an apple flavor. Actually, I think we already have wrapped it up, so it should be okay. You want me to put on this?
Right, so we're getting prepared to sample a C pen in front of us, and I'm going to be signing it off and passing over to Emily Cowell from Temple University, PhD student, soon to be Dr. Emily Cowell. Not soon enough, but yes, we all know that phase of the PhD.
Yeah, yeah. It's kind of.
Hello.
What do you have just down? Yeah.
Hi everyone, so we've just done a shift change, as Rachel mentioned before she signed off. We're just waiting for the sediment to clear and then we're going to sample this crinoid, I think it must be. Oh, it's a sea pen? Oh, cool, sea pen. It's hard to see through the sediment right now, but sample this sea pen. We'll be putting it into the quiver, and then after that we will be doing a biology transect between this waypoint and the next one.
try to find that part of the blade that touches.
So the intention of the setup of this manipulator arm is it's got a sharp sort of blade at the back that you can see there that's used for snipping the stalk, and then those kind of squishy fingers are used to hold onto the fragile parts of corals, but sometimes it's quite tricky depending on the texture of the organism. Sometimes they're very difficult to cut. I'm just going to go for the whole organism instead. Will that still fit in the coral quiver, do you think? Let's see.
Tail coming out.
Mm-hmm.
Does that matter? As long as the top one is safe inside.
It just came out.
It wasn't possible to cut it, so
Yeah.
I didn't fit
Mm-hmm.
I don't know if it matters, but you can put the head inside at the bottom and then cover it, even though it's squash or you cut it.
Yes, it's really big. If there is
Auto heading's not on. He has forward motion. He has forward motion. He has hand mic.
Just want to build it up, you don't want to lose the skeleton.
I really love to cut the bottom.
So we're just trying to decide where's best to put this organism, if it'll fit in the coral quiver or not.
Use the bio boxes. We can ask if we can use the bio box or one of the bio boxes.
What he can also do similar is he'll put the head inside at the bottom and then he'll put the cover in and he'll just tear it out.
But we will lose the other part, you know?
No, he can fold it in if you want.
Fold it in.
Yeah.
I could be like that.
Is it good for you?
Let's wait for her
Okay.
to answer if it is possible to use a bio box. If not, we will do that. That is better than just to have a piece because we can analyze the part that goes inside the segment.
Mm-hmm. Okay.
It's
He can,
better.
he can definitely fold that and put everything inside.
Okay,
Yeah, it seems very flexible.
if you can do that, it's okay because it is already
You
kind
already
of.
got it?
Oh,
Yes, okay, perfect, no problem.
she didn't reply yet, but
Okay, see here that is
said
okay.
we're folding.
Problem solved.
Yep. False alarm.
He already has the inside. He's just gonna get the bottom now and stuff in.
Yeah. Brilliant.
Basically fold it like this. Hold on, let me give you some better shape.
So the visibility is just a little tricky right now, so we're just adjusting some of the cameras to get a better view.
Yeah, it's going in. going is going in. Like when or we're gonna put it on.
Brilliant.
You're right in the middle of it. In a minute, it's coming out. It's coming, yeah, in the other side. I don't know if it's better. There you go. Fold it back in.
Oh yeah, fold like the top over.
Yeah. There you go. You have good sit camera.
Just make sure the bar doesn't.
Roger that. Watching it. There you go. You got it started. Oops. Watch it. Watch it. Come on,
Oh.
come on, come on, come here. You know what? Beautiful job.
Now just need to re-grab it on the cake.
Roger. No, I think if you fold it, it should fold like a cake, right? You could just stuff that around it.
After I thought every time I go like this, I was like, oh, it's
Yeah.
sitting.
Yeah. You know what? I could hold it down there.
Yeah, yeah, yeah. Yeah, we can get the whole thing in. Then I'll grab it on the inside. I'll flip it over now.
Looks like we're using some teamwork here. We're going to use both of the manipulator arms.
I'm gonna grab it. I'm just gonna hold on.
Great.
Okay, try that.
Get it loose.
I think we grab the tip on the end, and guide it in while I hold this puppet around.
Now, because if I would do this again, it would tell you.
Pushing it out. Go ahead and fold that over.
Oh,
Now you can grab from the top. Once you grab it, I'm gonna let go, right?
Yeah, just actually just keep your own nose. Actually,
Got it?
yeah, just make sure how this is going to move.
Let me get my arm out. Okay, it's open.
Well, we'll get up.
Come on up.
Yeah. Here on the bottom box. There you go. Nice. That was cool. Okay?
Okay, good. Thank you. So difficult, huh?
Yeah, they're those are hard
Yes,
to find.
yes. Flying away.
Can we zoom in a little more?
Nah, it's just an big signal actually.
This is like thick.
There you go.
Is it close already?
Oh, so it's in the last
What's the rope from the other side?
No, it's in there? Yeah. No, it's over here.
Yeah.
Yeah, it's just the stalk. That's holding it up. I think it's okay.
All right. It's not gonna go anywhere.
Yeah, so the head is in there, it's just the stalk is pinched in there.
Thank you.
Great. Well done.
And the gravity's
That
coming.
was a tricky one.
No por chin. You fix it.
Okay, thank you.
Grab it first.
Thank you.
It's por chin.
So we're just retrieving Excalibur, which is the probe for the heat flow. And then the next plan of action is to do a biology transect from here up until the next waypoint. So the settings we have for that are 0.2 meters per second for the speed, meter of altitude above the seafloor. We have the lasers on and we don't pan the camera or zoom on anything. And it allows us to do sort of transect where we can then later on count densities and have a sort of more quantitative sample of what we're seeing. Yes. Thank you. I think close enough. There we go. Perfect.
Bridge, ROV, that's an updated waypoint for you.
You want lasers on like that?
Yes, please. Yeah.
Thanks.
So do you have sort of autopilot settings that allow you to do that? You're just controlling it with your mind. Okay.
Yes.
But does it keep altitude as well? Oh, nice. It's a handy piece of kit.
Sometimes it doesn't work very well.
Okay.
Like right here.
Always trying to go miles away.
Let's see if that works. Doesn't work very well. Altitude. I'm gonna have to do it manually.
Okay. Does it tell you the altitude there? I see it. So if we were to see anything really interesting as we were going along on this biology transect, we wouldn't stop and we wouldn't adjust the camera, but we can drop a little sort of marker or take some screen grabs. That would mean we could come back after we've completed the transect. the sea cucumber that we're flying past here. There's been some interest in sea cucumbers on this cruise so far. Some of our colleagues are interested in looking at the microbiome of the hindgut of sea cucumbers to see if they have any microbial symbionts. So we've collected a variety of those so far. Little Galatheid off to the right there. A squat lobster. This will end up being about a 100-meter transact between waypoints 5 and 6. We've already done 2 other 100-meter transects by the looks of things. So that should be really useful for quantifying the densities of organisms that we're seeing. If you don't keep these kind of constant settings, it can be very difficult to get a good, more quantitative sample, because you want to calculate sort of how much of the seafloor you've actually observed in a transact. And so keeping these kind of set heights from the seafloor and angles on the camera really help with being able to do that. Sea urchin there. We collected one of these sea cucumbers yesterday. It was surprisingly gigantic when we got it up onto the ship. We were all quite shocked by the size of it. Really interesting organisms though. I've been really impressed by the variety that we've seen so far. Sea cucumbers don't get a lot of love usually, but I've been impressed. Another sea cucumber to the right. Some brittle stars too, these ophiuroids. They're pretty cool. They sort of drag themselves around the sediment with their arms, unlike sea stars that use their water vascular system, their tube feet. We've actually seen some brittle stars kind of taking off the sediment and going for a little swim too.
we gonna do a heat probe?
Yeah, if it's still looking sedimented enough. So we're just coming up to our next waypoint here. I'll try for a heat probe again. Yeah. Mhm.
Very deep though?
Yeah.
Turn around.
Yeah, we've been turning 90, but it's still there.
Yep. So we're going to attempt to do another heat flow measurement here. So it's a little bit of a slope that we're on. So they've been turning kind of 90 degrees to the slope to help with that. So we'll do that now. And then the first plan of action is just to get out the probe itself. It's about 1 meter long, and all of the probe really ideally needs to be in the sediment. So we'll do some investigation to see if the sediment looks like it's still deep enough here. So the sediment in these areas is really, really fine. And you've got very low currents. So often once the sediment's been kicked up a little bit, it takes a second to clear and give us a good view of what we're doing. So it's tricky to see right now, but that's the heat flow probe that we're just testing the depth of the sediment with.
Yeah, that looked pretty good, Rob. Yeah.
Cool.
Okay, we couldn't see it, but we got it all the way in there. So.
Great. So it looks like the sediment's deep enough here for heat flow measurement.
Okay, we're just gonna let it clear out just a little bit, just so we can see the probe there. Okay. Just because I think because we're so high up the slope, and we did see some rocks, probably not a bad idea to have a good eye on it. It's a good idea.
So we're just going to let the sediment clear for a minute before we do this heat flow measurement. Our interest in doing these heat flow measurements in sort of lines leading up to the seamounts and different features that we've been looking at on this cruise is to see if we can determine whether any of these features are either discharging or recharging sites for fluid. So a discharging site would be something, for example, like Dorado Outcrop that had the octopus nursery on it. They are showing usually higher than expected temperatures for the heat flow, and it indicates that slightly warmer fluids are being discharged from those sites, fluids that have been contained within the crust. Whereas a recharging site would perhaps be some of these larger seamounts, where that's one of the questions, and that's an area where the water is actually recharging into the sediment, into the crusts. So the reason for doing multiple transects sort of towards multiple points in a line towards each of these sites is to see if the heat flow seems to be increasing or decreasing as you come up towards the feature. And so on.
Looking pretty clear.
Ready?
Just 2 seconds. Okay, I'm recording.
Yeah.
So the process now, from my memory, is that we wait for 7 minutes, I think it is, and during those 7 minutes we're watching the sort of curve of the heat flow, the decay of the heat flow, and then it sends a heat pulse into the sediment after 7 minutes, and then we watch that for another 7 minutes and watch how that decays. And then lots of people who are much more clued up on this than me and go and analyze that data, do a lot of adjustments, and then we'll eventually be able to determine whether it looks to be higher or lower than expected. So our plan for after this heat flow is we'll head off to waypoint 7, which is another 100 meters up towards the slope of this seamount. We won't be doing a biology transect during that 100 meters, so we will be able to pan around and zoom and look at different organisms. So we'll see what we see, and then at the next waypoint we'll attempt to do another heat flow measurement. It gets trickier as we go up the slope because we know from yesterday's dive that we reach an area that's not very sedimented and it's got a lot of rocks. We don't want to break the heat flow probe. So if that's looking to be the case, then we might not do any heat flows there, but we'll see how it goes. And then ideally we might try another transect after that. But once we reach that kind of rocky area, I think our main focus, rather than heat flows and transects, is going to be focusing on switching to exploring for signs of venting, and following that depth where we sort of found the rocks. So yesterday we noticed the rocks seemed to be in a kind of band around the edge of the seamount. So instead of going up over the band of rocks and onto the top of the seamount, we'll follow the band kind of around the side of the seamount, see if we see any of those signs of venting. So that could be breeding octopus, it could be shimmering water flow, or potentially darker areas of rock where the sediment's been kind of blown away from the surface of the rocks. And if we see any of that, then we'll do some other things. So we'll be taking temperature measurements, potentially collecting rocks and water samples and various things. Pretty good, how are you? Something's sticking out of the sediment there. Maybe this, it's hard to tell, maybe the spines of an urchin or something like that. It's like some sort of big fish in the background, maybe some kind of grenadier. Again, we'll see if he comes any closer, get a good look at him. Looks very ghostly in the background. maybe some sea pens in the background there as well.
I'm gonna play with the camera.
Hmm?
I'm gonna let you play with the camera.
I always do it backwards. It always goes the opposite way to the way I expect.
So now you need it.
Because that's the zoom, isn't it? Yeah. Whoop. Huh? Oh, not a pen. Oh yeah, you can see the. Is that Octopus or they have that 8 it's on the collar? But this. There's different. So what would these guys be? Yep, correction. That's an octocoral. You can see the polyps there have the kind of 8 branches coming off them. Looks like another bamboo coral maybe in the background there. Whip coral.
Mind if I use it?
Yeah, go for it. Yeah. Yeah, so we're not doing the transact between this and the next one, so this is the next waypoint. Should we just want to put this up at the top?
I don't know because
It's damaged. But yeah, I mean, we could see if the second one maybe it looks like another one in the background there. Do you think?
I tried to pull around.
She want, Emily, did you say she wants a piece of that one?
Yeah, except that we would normally take a little snip off the top, but the polyps look like it's missing off the top of that one. So I don't know whether we can see if the one behind is any better before we move off. So for anyone that's just tuning in. Oh, is that where it's 8? Well, it claims there's a waypoint 8 on here.
It does. And there is.
yeah, for anyone who's just tuning in right now, we're doing some heat flow measurements. So that's what the manipulator arm is doing right now. Ooh. And we've got the heat flow probe in the sediment, so we're just waiting to get all of the data from that. And then after that, we'll be moving on towards the next waypoint, which is a little further up the slope of this Caballito Seamount. There's also some interest in, it's difficult to focus on them because of the sediment in the foreground, but there's some interest in potentially these whip corals. The one closest to us seems to have polyps missing from the top, and we would normally just snip a little section of the top, so we might potentially see if the one slightly further away looks any more promising. And then after that, we'll just be moving up to further up the slope to the next waypoint. And if it's still sedimented up there, we'll be attempting another heat flow measurement. But eventually, as we go up the slope, we'll probably come across this kind of rocky band around the side of the seamount that we came across yesterday. Once we get there, we'll change our priorities away from this heat flow and biology transects to following the rocky band around the edge of the seamount and seeing if we see any evidence of fluid flow.
We're all done. Thank you.
Nice. Can I have a quick zoom on? Yeah, I think that. Yeah. Yeah, I think that looks like it's missing a lot of the polyps. Sorry. Yeah. So if that's missing a lot of the polyps, do you want to look at the one behind? So that's just the heat flow probe there, we're just extracting. Then we're going to move off and have a look at these two corals in the background and see if we can potentially take a little sample off the top. We'll have a look at them. One seems like it's missing a lot of the polyps from the top, so possibly not that one.
You want the other one, right?
I think that one looks like it's missing a lot of the polyps. So yeah, if we can look at, I guess fly past it and look, but maybe have a look at the back one, and then get back on course.
This one is not this works.
Oh, it works.
I think that
Yeah, you think this second one's worse? Okay. We're thinking this this left-hand one looks probably like it's got even less polyps. So maybe if we can knit back to the right one.
Right? Oh, there goes my mouse.
Might be a.
I mean, I don't know. Is it easy enough to do a quick zoom on the top of this left one just to check? But I don't know how easy that is.
The one on the left?
Yeah, just to compare them. But I think it looks pretty bold, I would say. Yeah, I think that's probably got even less polyps on it. So can we have a quick look at the right hand one again?
Go back to
Yeah, I'm expecting it to come running through the door.
Driving back to Michigan. I think that's right in front of you. I saw it as ticker. 178. Come back to Starboard.
I'm gonna blame this on Emily.
Where's your friend?
Well, you can blame
Like,
the
I'm out, dude.
you can blame the bull Carl.
There it is. See, I sit down for living so. You in there. He got up and it went. It knew I'm coming.
Yeah, I think that's the one with slightly more polyps. So you want him to jump in?
Ready?
Yeah, we can. There was two corals. Odo wanted to sample on the top of one, but they're both quite bold. They don't have many. This is slightly better, but the one in the back.
We've got a little bit of a handover going on. You might hear a bit more commotion in the background. We've got folks swapping for watch and also going to be going off to or coming from lunch. So you can hear a little more commotion here and there behind us as that sort of thing happens.
Corey, I didn't see you in the gym this morning. I don't know if I just missed you or you're people stats.
See me.
I see you.
In the gym this morning? Oh yeah, I was there. I was there. Yeah.
Morris.
I know.
I'm just saying that maybe we just missed each other.
Yeah. I've never been this constant in the gym. I'm surprised myself.
So you know, Excalibur. Don't turn peppery now.
Sweet.
Okay. They might want to snip this from top. Other than that, there's only one thing. This guy has been used. Just the chloroquine one, and that's everything.
Sweet.
And it's copper. You might get one more probe up at 7.
Yeah.
Like we got a probe, but just carefully land.
Alright. Yeah. Once they started to get
Cap still off on that.
Lasers? Lasers are on. I'll find them for you though.
There we go.
They are. What do you want me to put them?
Wow,
How's that?
good. Looks good. Bueno, buenas tardes, buenas noches, buenos días. Aquí está Jorge Cortés de la Universidad de Costa Rica en otra sesión de 12 a 1 de la tarde. Los voy a acompañar y aquí estamos viendo este octocoral que vamos a tratar de recolectar una parte. Y ahí vemos los camaroncillos que nadan y viven asociados a estos octocorales. Good afternoon, good evening, good morning. This is Jorge Cortés from the University of Costa Rica. I'll be here for the next hour. And it's a great view of this octocrowl. You can almost see the sclerites in the body, the little white sticks. I'm going to try to collect a section of this. And thank you to everyone sending message, questions, and telling us where you come from, you're connecting from. And questions, we'll try to answer that. Pues muchas gracias a todos los mensajes que envían, preguntas también, y pueden contarnos de dónde nos contactan. Hemos tenido contactos de todas partes del mundo.
How much? How much do you think we should get out of this?
I would guess that, like here.
They're there, so
Yeah,
we
so
want in
have-
the buy box.
But this, how much would that, do you think that is? Yeah, like about
that'd be
quiver,
like-
quiver side.
That's bigger than quiver. That's like quiver that big, which would probably be but your dog.
Yeah, the quiver, yeah.
If you remember the collection of the sea cucumber yesterday, or the collection of the whale, or the fossilized whale skull, the pilots are currently discussing with the scientists kind of the size options of the containers on the porch of the ROV. So they're using Excalibur here to measure up an appropriate section so that they can fit it into, I think we're looking at one of the coral quivers versus a bio box, but we'll see where it goes.
How bendy do you think this will be?
Like another 10 centimeters for this.
Isn't it
Like,
right there?
yeah.
All right.
Yes.
This is what we'll do.
Well, that's
We'll
almost,
open up a coral quiver, see if it
it's
fits.
almost half a meter. Huh?
Yeah.
So
That's
maybe
pretty
it
big.
should be at the base of this. Excuse me. Here.
Well, that's what
Yeah.
I was saying. No,
Yeah.
but the problem is that that's that size.
Oh,
And,
okay.
and the
Yeah, it's
and the quiver
Yeah.
is about that size.
I mean, if it's bendy, like we can just trash them in. If not, file box.
Should be around 30 centimeters.
Or two sections. Yeah, we can cut one, put it in there, one, put it in there. Whatever.
It can also be 2 sections.
Es que, que cortarlo aquí.
Sí, que agarre una parte con pólipos.
Ajá. Lo que pasa es que esto es como medio metro. O medio metro y el tuyo es así. Entonces la pregunta es si lo hacemos en 2 secciones o lo volamos.
Es que la parte de arriba ya se pierde. Entonces en 2 secciones.
I haven't heard anything about the whale skull yet, other than it made it up fine. As far as the sea cucumber goes, if you check out hashtag Octo Odyssey on Twitter, we actually there's some photos posted there. It made it up in great condition, as far as I'm aware, and just truly impressed by the size of that sea cucumber. I don't think anyone was expecting it to be as large as it is. The cameras here, it's often difficult to, oh, let me fix my camera here. It's often difficult to judge the size of many of the things on the camera. They're either much smaller than you expect or much larger than you expect, unless of course we have the scale lasers on. Or a big Excalibur, which is proving to be a great little measurement stick here. But yeah, if you're interested in photos or more information about this expedition, on Twitter we do have hashtag Octo Odyssey. Of course, Schmidt Ocean we're posting on our own Twitter, but many of the scientists are as well. So check out that hashtag if you're interested in more information. And if you want more in-depth information about the expedition, you can go to schmidtocean.org. And we have an expedition page there for the Octopus Odyssey, which also includes some blog posts written by the scientists on board.
Empty.
See if there's a plan, right?
Yeah, so
Two
we're-
cuts.
there's 2 bare spots. We'll cut it at the low and bare spot.
Yep. And then
Uh-huh.
a second one.
Yep.
Are we doing a transect to the next transect?
Yeah.
Whoa. That's not good. Put that back.
Yeah.
Put that back. And then you gotta put the other one back in there.
Yeah, that one doesn't
That
want
one
to.
wasn't
Doesn't want to leave.
put in there very securely, I guess.
Little snag with a previous sample there, so pilots are gonna restow that.
Did that float you guys think down? I think they had some of the same problems that we were going to be faced with.
Because it says sort of transit.
Yeah, this one is-
Okay, this one doesn't have a transit.
Yep.
Okay, I see.
Next one, but not this one.
Okay,
There you
good.
go, there you go. There you go.
Mm-hmm.
Come on. Alright, the most of this thing that's this thing is just I try to push it down with the cap on it. So I grab just the tip of it and kind of flip all the way down there.
Oh, a fun question here about how the pilot is controlling the arms, or the manipulators, you'll hear us call them. It's more than just a joystick. What Scott has in front of him right now is like a miniature model, a miniature articulated model of the manipulator. And as he manipulates that controller, as he changes the angles of the joints and rotates it, that actually is fed to the manipulator itself. Kind of a cool control system, but also can be a complicated control system. So our pilots have a lot of practice with them, but watching them do it is always just kind of like, you're like, boy, that kind of looks like it makes sense, but also at the same time, it's a lot to keep track of. Yeah, I'll see about showing you all that at some point, if I can get a camera hooked up for that.
Get closer in. Oh, we have it. Uh-huh. There.
Is that good?
Yes. Yes, perfect.
There you go.
Oh, that was clean cut. Nice.
I never got it. I had faith in you, really.
And make a note that he has hydroids.
I was feeling the love.
Okay, this is gonna be like that. Yeah.
What I should have done was gone 45 on it.
We collected this octocoral and apparently has some hydroids. Estamos recolectando estos octocoral y con unos hidroides, y en inglés explicaba sobre el brazo que se controla, hay como un brazo en miniatura aquí adentro en la sala de control con todas esas partes, y los pilotos que se han entrenado por años van moviendo esto para poder manipular ese brazo.
I've been a handballer before. Okay, so for this
Maybe.
one, just guide me up, down.
Yeah, maybe it's just
A little
like,
bit lower.
yeah, that
Keep
would
going
be like
down.
that would be like 10, 20, like, yeah,
There you go, right there.
like
Right here?
30 centimeters. So.
All right. Cutting?
Yeah.
for it.
Wow. So, yeah, some of this cut very nicely.
This is gonna be a little bit more of a push it down thing.
Yeah.
But again, it shouldn't float at all.
Escaping worm. Oh, yeah, yeah.
Yeah, you can see you're gonna push it down.
All right, cut it twice.
Maybe, yeah.
Yeah, if you want.
Am I allowed?
You mind if he cuts it a third time, second time? Yeah. Fourth.
Yeah. Let's see how flexible it is.
That's just gonna spring back. No?
Oh, no.
I would pinch it and push it.
Yeah.
Good.
You're right. Just perfectly blocked by the.
Yeah.
Got a pretty good view in the nip cam.
Go ahead.
There you go. Perfect.
Mm-hmm. I imagine it's
There you go. That's good.
Sweet.
Good job, man.
breaking, but that's okay. Mm-hmm. Great. Okay, great.
What's your name? Or ROV, no?
Yeah, so now we move on to waypoint 7.
Alright, I'll set us up for
Yeah.
the transect.
And we do no. Yeah, now we just move to waypoint 7.
Just no transect?
No transit. The
Okay.
heat flow, and then the transit.
Alright, so no transect this time.
No. Yeah, it's one with transit, one without.
Alright.
Todas las muestras que se recolectan van a ser depositadas en el Museo de Zoología de la Universidad de Costa Rica y se toman sus muestras ahora para análisis genético y se le hace también todo el estudio morfológico. Y todo esto se preparan publicaciones sobre todo este material. So everything that's collected goes to the Zoology Museum at the University of Costa Rica, and every sample there's a subsample for genetic analysis, and the rest is used for morphological analysis, for the description, for the identification of the species, for reporting, and if it's a new species, then the whole process starts to analyze in great detail compared to the original organisms used for the description, and then publish that.
Hey, on the bridge, got an updated waypoint for you?
What, Jason? Is that Tony? Tony.
So we're moving now from waypoint 6 to waypoint 7. We're not even halfway of our dive plan for today, so we're gonna just move over to waypoint 7. There we will do a heat flow measurement, and after that we'll do a transect. Ahora nos desplazamos del punto 6 al punto 7 para continuar la trayectoria propuesta para esta inmersión. Y estoy viendo que son 17 puntos en total y vámonos apenas llegando al 7, así que nos falta un buen rato todavía de trabajo. Vamos a llegar al 7, ahí vamos a hacer una medición de flujo de calor primero y después un transecto. What we're seeing is at 3,083 meters depth. Temperature is 1.84, and some more environmental data. Salinity is 34.7. Oxygen is 32.8%. Continuamos desplazándonos. Aquí estamos a 3080 metros de profundidad. Una salinidad de 34.67, temperatura de 1.84 grados centígrados y un porcentaje de saturación de oxígeno de 32.8%.
Sorry, I tried. Too late.